So we were discussing about the applications of PCR. Next is RAPD, random amplified polymorphic DNA. Random amplified polymorphic DNA. So PCR can be used to study DNA polymorphism. For that we are using a primer that is random. We are using a random primer for random amplified polymorphic DNA. So in this study we are using an arbitrary primer. That are arbitrary means we are selecting without any reason. We are selecting a primer of a 10 nucleotide primer we are selecting, and we are amplifying the DNA. So after PCR we are doing what? Electrophoresis. Then we will get bands like this we are getting. Then we will compare it with the known species. So we can study about whether the members are closely related or distantly related based on the banding pattern. So based on the banding pattern we are getting in the electrophoresis, we can study about the phylogenetic relationship between these species. So in RAPD PCR, the segments of DNA which are essentially unknown to the researcher. For us, the segments of DNA are unknown. We are using only an arbitrary primer for that. So this primer will attach or anneal to different sites or multiple sites in the genomic DNA. In the genomic DNA, these primers will attach to many sites. So because the primers are very short and they often anneal to genomic DNA at multiple sites, and the amplified DNA polymorphism is detected as distinct pattern after gel electrophoresis that we have discussed. So we can study about the phylogenetic relationship between the different species, which are, this one is unknown to the scientists. So we can identify an unknown species with this technique.